Hey guys, I'm back. I know it's been a while. I apologize for that since I've made one of these videos, and for all I know, none of you will even watch this one. But I know Trey will, so this one's for you, buddy. So you requested it, so I'm going to make one here. So we'll see if I remember how to do it. The chapter that we're doing, remember, is on DNA and the wonderful world of DNA. So we're going to go through this pretty quick. I don't think it's going to take very long. A lot of this you've already seen in class, but who knows, maybe you need a quick review. Remember, these two guys here are pretty famous scientists, Watson and Crick. They were the ones that discovered that DNA was in the shape of the double helix, and they came up with the shape of it. And there was a big race going on between them and a couple other scientists trying to figure out what is this DNA going to look like. They're the ones that they cheated a little bit, I guess you might say. I kind of think they looked at Rosalind Franklin's X-ray crystallography, and but they're the ones that they did have to come up with the image and the idea themselves, and they did that. And so they're the scientists that did that. We're not going to watch this video. You saw that in class, so let's go ahead and move on. If you remember, before Watson and Crick, there was a big controversy of what is the source of our genetic material. Is it DNA or is it protein? And the scientists in that day and time really were leaning towards protein, because when you looked at the two structures, protein have all the different three-dimensional shapes. They had more building blocks, because you remember 20 amino acids make up most of our proteins, and then DNA over here only had 4 building blocks: your adenine, thymine, guanine, and cytosine, and it's not near as complex. So everyone was thinking, oh, it's got to be proteins. Well, if you remember, the big experiment that I want you to really understand was one of Hershey and Chase's, where they used radioactive phages, the bacteriophages. And remember, bacteriophages are the ones that are viruses that will actually infect bacteria. So they grew these phages, first of all, in radioactive sulfur, and so that phage would incorporate that sulfur into all of its protein. And remember, the part of the virus that's made of protein is that outer coat and the outer shell of it. And so they allowed them, put them in with some bacteria, some E. coli, let the phages infect the E. coli, and put them in a blender, agitate them, separate them from each other. Then they put them in a centrifuge, and they found that all the parts of the bacteria ended up at the bottom of the test tube. And with the radioactive sulfur, there was no radioactive sulfur in that pellet that formed at the bottom. So that told them that phage did not transfer its genetic material through the protein to the bacteria. So they set it up again with the radioactive phosphorus, which we know phosphorus is pretty commonly found in DNA, in the backbone of this DNA molecule. Did the same thing. This time the phage actually incorporated the phosphorus into its own DNA. It injected it into the bacteria, ran it through the centrifuge, and then so that radioactive phosphorus then showed up in the pellet. So that told them that hey, there's that phage transferred its DNA into the E. coli bacteria cell. So that was again Hershey and Chase, and that was kind of a way we came up with finding out that it was actually DNA that carried your genetic information. Chargaff, remember Chargaff's rules, I want to go over real quick. Thymines are always going to equal adenines, and cytosines are always going to equal guanines. And you can use, if you have the percent of one, you should be able to find the percent of all 4 of them. We went over that in class. Remember, if you have, let's see here, I can write here. If you had, let's just come up with 40% adenines, that tells you that you have 40% thymine. So that's a total of 80%. So that leaves you with 20% of cytosine and guanine. So how many is that going to leave you? That will leave you 10 with cytosine and 10 with guanine. So again, that's Chargaff's rules, and you should be able to be able to do that for the test. Let's move on here. Go ahead and talk about real quick the whole anti-parallel conformation. Remember, the 5' end over here runs 5 to 3, and then the other one's going to run 5 to 3. Where's my mouse? 5 to 3, and then 3 to 5. So that's anti-parallel. And if you remember that the new DNA molecules, this is the growing that these. Get this mouse over here. It doesn't want to cooperate with me. Remember when we're getting ready to talk about DNA replication, the polymerase 3 can only add the DNA to the 3' end. So that's kind of the growing end. So it's only going to be able to add it to this end of that strand. It cannot add it on the 5' end. So let's move on here and talk about DNA replication, and we'll be done with this. I encourage you to go back and look at some of those videos if it might help you online that we looked at in class. Again, just search DNA replication and you'll find tons of little animations and stuff, and go to YouTube and you can find them as well. Real quick, before we run out of time here, with DNA replication, this is the original strand. Remember, it's uncoiled by this little protein here. This enzyme is called helicase. It's going to unzip it and uncoil it.
We'll see the remarkable
Same
way
time.
our DNA is tightly packed
Hold on.
so that 6 feet of this long molecule fits into the micro.
Okay, I guess she really wanted to talk. Sorry about that. So that leaves you with the the strands open up here. This first strand here is going to the DNA polymerase is going to add the new DNA on as it opens up. It's going to add it. Remember, that's the leading strand. It's going to add it in to the 3' end. So it's adding it to the 3' end. So if this is the original strand is 5 to 3, that means this end of the new strand is 5 down here and 3 here. So it's adding it to that growing 3' end. Down here, remember this strand, it can't work that way. It still has to add it in that add it to that 3' end, and so it has to run in the opposite direction. And that leaves you with these little segments that it makes segments at a time. It can't add it in the direction of the replication fork. So you've got the different enzymes involved. The primase lays down a primer, and then it's going to go ahead and fill in the gaps there towards the previous one. And once it's completed, once it's connected, the DNA polymerase 1 will come in and remove the primer and replace it with DNA, and then ligase is going to come in here and seal them up, kind of connect the two together. A couple other of the enzymes that Warren mentioned. Remember, the topoisomerase is the one that's going to be down here to help relieve the strain. And what is it? The single strand, what's it called? Single strand binding protein. I don't want to get that wrong. Is what's going to keep, I think it's what these right here are, it keeps it from reattaching to each other. So that's a little bit about that. Look at the little picture right here on the in your book too, and I think that's a good thing to look at as an overview. To remember the origin of replication, you've got the replication forks on either end, and your leading strand and lagging strand. And so we've gone over this a bunch in class, so I'm hoping you have a pretty good grasp of it at this point. The only other thing in this chapter that we talked about proofreading and errors. Remember, nuclease is the enzyme that will cut out when there's an error has been made. It will cut out the little section, and then the polymerase will come in and add the new DNA. And we also talked about how the problem that we have with our cells with the telomeres at the end, because you're going to get shorter and shorter and shorter strands of DNA after each replication. And so to prevent a couple of things that we've done, that your body has done to solve that problem, is we have the telomeres at the end, which are just repeating subunits that don't really code for anything. And so that will allow us to get a bunch of replications and not damage any of the vital information in the DNA. And the other response your body has is with telomerase, and that is an enzyme that's used in your in the making of your gametes, so that your gametes are always, through meiosis, whenever, even remember in meiosis the cell replicates, all the DNA synthesis takes place too. And so this telomerase will actually fix that problem and add the new DNA in for each time before each time that DNA is synthesized, so we don't have the problem of it shortening each time. So that's in a nutshell, that's that chapter. I went through it pretty quickly. Hopefully maybe I said something that you were confused on. If you have any questions, ask me in class, and we will see you guys in class. Hope you enjoyed the video.